All right. So here in the CNV workflow, you see many components. They are modular. In white, we have inputs and output files. In gray, we have functionalities that is handled by the tools. And here we have boxed two of the functionalities because this is what lies at the heart of CNV, our CNV workflow. Now remember that I mentioned that the somatic SNV and indel discovery workflow doesn't call loss of heterozygosity events. The CNV workflow is the basis for the allelic CNV workflow, which can detect loss of heterozygosity events. And even though I don't show a diagram here for ACNV, we will cover it at the very end. So let's walk through some of the concepts behind this workflow, and then we can get into the details of each of these steps. This is just a reminder, and I just showed this like 20 minutes ago, that copy number alterations must be taken into account in conjunction with short variants. And you can see how dramatic somatic copy number variation can be. These cells are from the same individual, and they've been made into cell lines. This is the matched normal for a breast cancer cell line called HCC 1954. And here we've painted each chromosome with a specific color. In yellow, we have chromosome 1. In red, we have chromosome 2, and so on. You can see for the cancer cell line that we have 6 copies of chromosome 1 and bits of other chromosomes fused onto the ends of chromosome 1. Same for chromosome 2. This cell line is very aberrant, and this can happen in cancer. Alterations vary dramatically between cancers and even within cancers. So we don't just care about copy number variants for the somatic context. Someone made this slide and listed highly amplified and deleted genes in cancer, but we also care about copy number variants in the germline context because they are involved in certain conditions and diseases. And so I know I'm focused on the somatic workflows here, but I just wanted to point out that we have a germline CNV caller that's in beta. It's in beta because it's still being worked on, and it is only available to run in Spark. And of course, all of our workflows are near to completion for targeted capture data, but for whole genome-seq data, they are all under still experimental status. So the somatic CNV workflow is ready for your use. The allelic CNV workflow, if you look at the list of tools, they are out of beta status. They look like they are ready to use, but in fact, the workflows themselves are being, our developers have taken an interest in improving the workflows and the underlying math. But if you need allelic CNV analyses now, I would use these. They really improve the results of CNV. And okay, many of the tools between targeted capture and whole genome seq are shared, but of course the workflows are different. And this chart I'm showing you, this table I'm showing you just to give you a sense of the different tools and workflows. And the one thing I really want to point out is that to do any copy number variant analysis, you need to get read counts. For whole exome or targeted capture, you use this tool called calculate target coverage. And for whole genome, you use Spark genome read counts. And here I'm also listing the inputs, but I will not get into those. You have an option to correct for GC bias in read counts with a tool called correct GC bias. And then we have the somatic CNV workflow, which takes the read counts or the GC bias corrected read counts and has 5 tools you can use to get copy number variant calls. And then the allelic CNV workflow builds on that, takes the outputs of the CNV workflow as well as a matched normal BAM to give us allelic CNV results using these 3 tools. And these are the only 3 tools that are actually out of beta status. There are a variety of tools available for allelic CNV, but these are the main ones that we recommend. So we make an assumption, and that is that copy number variants are proportional to coverage depth, that they will alter coverage if the number is altered. For example, here, for normal tissue, for a control locus, you have 2x coverage, and for your TP53 locus, you have 2x coverage. And so you would say, for this diploid context, that they have the same copy number or copy ratio. But in a tumor, we would expect, if there is a copy number alteration, for that to be reflected in the coverage depth for that locus compared to other loci, plus some mutations probably, because this is a tumor suppressor. And here we have 5x. We also assume that there isn't bias towards sequencing contexts and sequencing methods. But if there is any bias, if there is any inherent bias, this bias will be systematic. And we can account for this bias. So what do I mean by systematic bias? Well, if you take different sequencing technologies, you'll see that coverage is variable for whole exome targets and kits. And here I'm showing 2, ICE and Nextera, versus whole genome sequencing, PCR-free here. What you can see is that the coverage charts, which are at the top of each box, are different. The whole exome capture gives mountains, whereas the genome, whole genome gives what looks like maybe an ocean. So the thing about exome capture is that there is a step involved that uses a bait to pull out the inserts of interest. And this bait can be RNA or DNA, depending on your capture kit, and can affect the representation of coverage for the different targets. So it's important for our normal control to come from the same capture kit and have the same capture targets as the tumor that we want to analyze. And this normal control should capture sequencing artifacts and noise. And because sequencing artifacts and noise have some random element to them, I think it would be better to have a number of normal samples to capture that. And so our control wouldn't just be a single normal sample. Our control would be a panel of normals, or a PON. So an important limitation of our method is that we are looking at copy ratios, not copy numbers. Let me explain. A copy number is an absolute integer valued number of copies for each locus. For these 4 different cells, the copy number would be 1, 2, 3, and 4. The copy ratio profile is relative ratio of number of copies of each locus to the average ploidy within the cell. And so each 4 of these would have the same exact copy ratio at 1. Okay. So here are some raw copy ratio profiles from exomes. You can see that they are extremely noisy. I mean, would you say that there are any copy number events here, just by looking at this? No. Yeah. Okay. So it's hard to tell if there are any events. So we're going to resolve this, and you'll see that there are actually events in this tumor sample using the CNV workflow. So in step 1, and I'm going to go into detail for each of these steps and give you the commands. But in general, we take BAM reads and our targets, and we put them through calculate target coverage to get proportional coverage numbers. These, for our normal samples, we feed into two tools to get the panel of normals. So then once we have a panel of normals in step 3, we can take our tumor sample and normalize the somatic read counts to get two different files. One is called a pretangent normalized coverage, and the other is called a tangent normalized coverage. I will explain these in a little bit. And in steps 4, and in this step here, we can perform segmentation and call segments. So after step 3, what we have is an individual row for each target and an associated copy ratio. If you have 20,000 targets, you're going to have 20,000 rows of data. That's not what you're looking for in your segmentation. You want to group regions of contiguous regions of the same copy ratio into a single segment to get a file that represents the actual events. And that's what segmentation does. And then you call on those segments, whether they are amplified, deleted, or neutral in terms of copy number. And we have an optional plotting step, which we highly recommend. And all the plots that I'm showing you come from this plotting tool, and it also emits some QC metrics. Okay. So step 1, we take our reads and our targets. We go through calculate target coverage, and we get our proportional coverage profile. There's 3 things to remember here. One is that you take your bait or target intervals that the capture kit provider gives you, and you want to pad them. You want to pad them. We recommend 250 base pairs on either side. We have a tool that does this called pad targets, and it will intelligently pad them. Because in our hands, this has given us better results. You want to make sure, too, that you collect proportional coverage, because this tool defaults to raw coverage. So you have to specify proportional coverage here. And third, you want to disable the duplicate read filter, because we want to include duplicate reads in this analysis. Because in our hands, empirically, this has given us better results. So after this, if you want to do GC bias correction, you would do it right after this step. So the reads that are counted overlap the target. Even if it overlaps a little bit, as these purple ones do, they're all counted. The green ones are not counted towards the coverage. And for proportional coverage, each target's read count is divided by the total sample reads for that sample, and we get our proportional coverage. In step 2, we generate the panel of normals. Here in this diagram, I'm only showing 3 normal samples, but this is just a toy example. We typically recommend at least 3, if not 40 or more, normal samples per pawn. For certain capture kits, we found 150 to 300 normal samples is where the benefit plateaus. So you want to do some testing for how many normals you will need for your pawn. You put them through a tool to combine all of their read counts into a single file with combined read counts, and you put that combined read counts file into create panel of normals to get your panel of normals. This panel of normals is in what we call HDF format, hierarchical data format, and you can actually explore the data in the pawn using this external tool called HDF view. And I don't think I need to go over the commands. They're very straightforward. Now, this is the heart of the workflow. We tangent normalize our tumor coverage. We take our tumor coverage profile, we take our pawn, and we run it through normalized somatic read counts to get a pretangent normalized coverage that has been normalized against the pawn's median, and a tangent normalized coverage that has been normalized to the pawn's principal component, which is the systematic noise. And here I'm showing the results of running those steps. Here we have our raw copy ratios, and we remove noise by target medians. We normalize by target medians. We see some events appearing, and then we normalize against the principal components by tangent, and we see even more resolution of events. And here the x-axes correspond to genomic loci, and the y to copy ratio, where each dot represents a target. And you can see this fuzzy caterpillar has become slim and has resolved events. That's not the end of the story, because you have, what, 20,000 rows in your file? You want to segment and call copy ratio events. And so you take your tangent normalized coverage to perform a segmentation, and this produces segmented coverage. Then you take, again, your tangent normalized coverage and your segmented coverage and run them through call segments to get your called segments. So again, you're grouping contiguous targets with the same copy ratio. We're consolidating, and when you call, you are making one of three calls: neutral, amplification, or deletion. So to indicate segmentation, we alternate the color of the segments between blue and orange, and you'll have a file that outputs for each of the segments a call 0, plus, or minus. And I think you can figure out what they mean. Here is a example call file, and the segmented file is identical to this file, except for in the called segments file, we add this last row. And in the last step, you can plot. Plotting takes the pre-tangent normalized coverage, the tangent normalized coverage, as well as the segmented coverage, and gives you plots and QC metrics. And in the alpha version of this tool, we didn't have a sequence dictionary requirement, but now we require this, which means that you can plot against any reference. It doesn't have to be B37. That was a limitation of the alpha version. And depending on how you order the contigs or the representation of the contigs in this reference dictionary, plotting will just follow in order. So you can tweak the plots to what you want them to be. So you might think that the median normalization is sufficient to see some events, but you want to do the tangent normalization, because you can see here median normalization did not resolve these rather peculiar events. I'm not sure what's going on here. Something is very strange for this chromosome. You have scattered amplifications and deletions across this contig. Okay, we have 10 minutes to cover ACNV. So allelic copy number variation has, again, many associated tools. Most of them are still in beta and experimental status. The tools that are good to go are listed here. There are 4 of them: GetHet Coverage, Allelic CNV, Plot ACNV Results, and Get Bayesian Het Coverage, which is a different way of getting het coverage using different masks. So our developers have plans to improve upon these methods, but if you need to do ACNV calling, you should be able to use these tools. And running your results through ACNV will greatly improve your results. And the one thing is that you need a matched normal. You need a matched normal to do ACNV. And you need the results from GATK4 CNV. And you will detect 2 things: the minor allele fraction and any loss of heterozygosity events. And so if you have a diploid cell, and let's say a gene with an allele A and an allele G, it's heterozygous. But let's say that the allele A contig duplicated in the cell, and the cell actually has 3 of this contig. So here, there's 2 A alleles and 1 G allele. G is the minority here. So the minor allele fraction becomes 1/3. And that's something the ACNV workflow plots. Now say then that the config with the G is deleted from the genome. And all you have left are 2 contigs with the A allele. Your copy number is the same as your normal diploid cell, but you've lost heterozygosity. And this is what we mean by copy neutral loss of heterozygosity. And these events occur more often than you think in cancer. So just briefly, the tool uses something called heterozygous coverage, or HET coverage, and counts the tumor alleles for normal heterozygous sites. So first, you collect the ref and alt counts at common SNP sites in the matched normal. And then you perform statistical tests at each site and filter on heterozygosity. You only want to have normal heterozygous sites that are fairly confident in terms of their heterozygosity. And then you take those sites and you look at them in the tumor and you collect ref and alt counts for the tumor sample at these sites. And I think you can understand how this works underneath the hood. And so if you apply ACNV, here are your results from the CNV workflow, and you can see how ACNV cleaned up the segmentation, fused a lot of the segments that were previously noisy, and I think reduced the amount of noise in general with this cleanup. And you get this plot of minor allele fractions where you can see that mostly the cell is diploid. It is maintaining, well, diploid or tetraploid, maintaining heterozygosity. Here in the region of the amplification, we have a minor allele fraction that likely corresponds to 1/3. And here we have a loss of heterozygosity. And what we see here probably is normal contamination of the tumor. So here's an interesting case. Here's segmented copy ratios, and it looks like this cell is mostly normal because the copy ratios are at 1. And that we only have like maybe 2 amplification events. It doesn't look all that aberrant. But if you look at the minor allele fraction, you see that most of the genome has lost its heterozygosity. And in fact, the 2 amplification regions are what have maintained their heterozygosity. So highly recommend augmenting your CNV analyses with ACNV because you can resolve things like this. All right, that's that. If you have any questions, I will take them.